Hi guys, welcome to Genetics Junkie and my new video on gene order and genetic distances. This video in particular is about no double crossovers. So I just want to let you guys know that this question that I'm doing is actually from our TUT 5 and it's question 1. So you guys can pause the video and get that out if you'd like to. If you don't have it, then the question is in the comments, so you can take a look at that. Alright, so for this question, let me just get it on my own computer. Okay, for this question, they specifically want the gene order and the map distances between the three genes, right? And what they've given you is the F2 offspring, and they've also told you what the F1 parents look like. So before we look at all of this, I just want to explain it to you. So right at the bottom over here, I've just shown you that my pluses are for all the wild type. So those are the dominant phenotypes. And then for these ones, these are the lowercase letters are my mutant recessive phenotypes. And they also tell you in the question that there's three genes. You got one for forked, one for outstretched, and one for garnet. So yeah, that's what my F, O, D, and G mean. I want to just bring it to your attention that I've written out all of my F1 in symbol format. In the question, they just write out words and they give you the totals of each of those classes. But here I've written out the symbols and I'm going to explain to you why. Also, I've written over here that there are three pairs of F2. So it's 1, 2, and 3. Three pairs, obviously 6 individuals, I should say. Okay, and that should show you that there are no double crossovers involved in this question. If there were 4 pairs, then you would say, okay, double crossovers are present. I've also written all my offspring in, I would say, descending order here, so from highest to lowest. Remember, you have to put them in order. And that's the first thing you do when it comes to doing your gene order and you have the F2 progeny. So the highest numbers, always remember, are your non-crossovers. They are your parental types. So they will always be the highest. They tend to be more than 50% of your total progeny. And then anything after that are your recombinants. And here's my single crossover 1 group. They are just below the no crossovers. And then my single crossover 2 is usually the least. In this case, there's no double crossovers, but they would be the lowest group, lowest frequency, I should say. Okay, I also wanted to point out to everyone that some of you may have missed this out, but they say in the question, they say that your wild type F1 females are heterozygous at all 3 loci, and they're also crossed to recessive males. So obviously a recessive male, this is for your F1 by the way, you don't have to write it out, but I want to because it kind of is going to show you a little bit of a trick, right? So I'm going to write my F1 out over here. So they say that the males are all recessive, so I'm just going to write that out. So they're obviously recessive for both of those genes. They're also recessive for both of those genes. And they're also recessive for both of those. Right, so that's my recessive individual. But now they say in the question that there's a female that's wild type and heterozygous for all 3 loci. And now you could be asking yourself, okay, well, how is it written? So all you have to do is look at your non-crossovers, because your non-crossovers look like your parents. So it's literally those 2. So we can just write that out. Cool. So that's a trick that you can use. Just remember your non-crossovers and your F2, they look like the parents. So just take that into account. And remember in my last video, I've got new highlighters now, when we're doing our parentals, like our parental group, we always take the top 2, and we take the bottom 2. So if we do cross them, that one and that one looks like that one, and that one looks like that one. So it's very easy. Okay, and I just drew a arrow pointing there so that you know, like, I'm moving in that direction from left to right, as I stated in my previous video. Sorry. All right, so the reason why we write down our symbols is that we're going to use them in what is this called? In our gene order, right? So when we've put them all numerically in order, you have to see, okay, what's in my no crossover class. And for this particular example, they said that the first individual is just outstretched, and that the second individual is garnet and forked. All right, so I've written it in this order, F, O, D, G, because in the question, like in the actual paragraph, they first present you with forked, then outstretched, and then garnet. It's in order. So then I'm like, okay, well, I'm just going to do it in that order first to start out with, because we're still determining the gene order. But the problem is we don't actually know what the gene order is, but it's a good starting point. So in my opinion, you should just start out with what they give you, sort of. I was checking that my video is still going. Last time it switched off, and then, yeah, this is the second time I'm doing this. I was really bummed. Anyways, right, so for the first individual, they say they're only outstretched. So you know that it's wild type for the first gene, because it is not recessive, but it does contain the outstretched mutant, so I have to say it's OD. And then for the last one, which is garnet, it is also wild type. We get that from our F2, from the F2 frequencies, right? So if you look at that, you'll be able to see the corresponding letters. And you do that for all of them, right? Just remember, when it doesn't tell you, if it says it's just forked, for example, like in this case, just remember it's forked only for the mutant recessive trait. Not for these two, I should say. They're both wild types. So just remember, like, they're not trying to trick you. It's just that they're not letting you know that they are wild type. Okay, so let's move on to the actual gene order thing. Right, so to distinguish between my individuals and my order is going to be in capitals, like I've written here. And then all my other little thingies, my gene symbols, are going to be either in lowercase or a plus for wild type. Now, like I said before, the reason I started out with F, O, D, and G was because it's actually in the paragraph in that order. So that's your first thing that you need to do. You need to write down your orders here. So I got my F, O, D, and G, and now I move on. I just move this one to the left, if that makes sense. So now each gene needs an opportunity to be in the middle, if that makes sense. So in this case, where OD was in the middle, OD has now moved to the left, G has been moved up, and then F obviously goes on into the right position. Now you need F to be in the middle, so you've moved everything up one. So now F is in the middle, G has been moved up one, and OD is now on the right hand side. So without looking at anything, you've just written that out. Okay, cool. So I've left this block open to be able to fill it in. So this particular area is just for my no crossovers. So now I look here and I go, okay, it's wild type for the F gene, it is mutant type for OD, and it is wild type for G. And for this individual, it is mutant type for the F gene, it's wild type for OD, and it's mutant type for G. Great, that's how I filled it in. But this is still the same as this. It's just that I've shuffled it around to be able to fit, to correspond to its heading. So this individual is still that individual. I've just moved the OD under this heading and made it plus plus there. And the same goes for this. And then obviously this also needs to correspond under its heading. So you just write it down. Then you must look at your single crossover. So I've done the first block. I'm just going to do fill in this block for you so you understand what I'm doing. So I look here and I'm like, okay, cool, it's got an F, and then it's plus plus for the other genes. And this individual is plus for F, and then it is mutant for G, and it's mutant for OD. I hope everyone's okay with that. And then obviously these two sides you do the same. You still look at your single crossovers and you fill it in. You still look at that, fill it in. Then you do your single crossover 2, and you go, okay, this is all plus, 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 plus. For all of those, it's all completely wild type, and this is completely recessive. So you're going to say G, F, and OD. So there we go. We filled out a complete table for our no crossovers, our single crossovers, and our single crossover 2s. So that is corresponding to that, that corresponds to that, that corresponds to that. I hope everyone understands that now. Okay, so now the next thing that we're going to do is we're going to compare our single crossovers to our no crossovers. So now I'm going to grab my highlighter and I'm going to just draw lots of lines all over my little picture here. So this is supposedly one of your crossovers. We're not sure which one, so I must actually assess it and see, okay, if I do this cross, will I be able to find a plus plus G individual in one of my crossovers? And it looks like I don't. So this is never going to be our gene order. So I can already cut it out, which is great for us. So now it just leaves us with these two, right? So we do the same here. Will I find ODGF in one of my offspring over here? So yes, we do. We actually see it here. I'm going to highlight this just to show that the cross between O and G gave me this individual. And then the cross between O and G again gave this plus 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 individual. But we look here as well and we go plus F plus. And this is found here. So now we have an issue where we actually don't know whether it's this one or this one. So we do it again. We do our other single crossover from the other side. And we can check again, same way. So we go, can we find a plus G plus in this individual? And no, we can't. So it will not be this gene order. Then we look here and we go ODFG. Can we find it here? And yes, we do. And if we go the other way, plus, 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 we find it here as well. So we know for a fact that this is now, this is going to be our gene order, which is great for us. So I'm just going to write it down here. Gene order. And we know it's going to be GFOD, which is great. So we've figured out our gene order, but now we still have to figure our distances between the genes, right? So now we're going to do a little bit of little calculations, I should say. So what you do is you need to, I should say, take your number of your total progeny in your crossovers, and you need to times it by 100 to be able to get your percentage. But before we start doing that, the reason why I marked these in pink is so that I can see for myself that this is my single crossover 1. So like I did it here. That's my single crossover 1. And what did it happen between? It happened between G and F. You can see it there. It happened between G and F, right? So I'm just going to write that down. And for single crossover over 2, it happened between F and OD. It's like that. And guys, just write all of this down. It makes so much sense towards the end. It doesn't make you confused. It allows you to kind of figure out where the single crossovers happened, and it just allows you to visualize what you're doing. All right, so now I go to my single crossover 1, like I wrote it down here. And I go, okay, the total progeny is 117. So I'm going to write that down up here. 117 over 1,000 because the total number of progeny, which I wrote here, is 1,000. So it's always good, like, do your different classes. So I have my 3 classes, and I wrote down all my progeny, but I also got their totals, and I got the total of the totals, if that makes sense. So, you know, it just helps you towards the end. If they're going to ask you about map distances, just get your totals because you will use it. Okay, so my single crossover, like I said, I'm in my gown, so it's late, whatever, is between G and F, right? So if I do that calculation, I've got it here somewhere, it is actually 11.7%. But remember, your percentages are also directly proportional to your centimorgan, your distances between the genes. So on our genetic map, well, we'll get to that. Okay, let's just leave that. So now we've got our distance between G and F. Now we have to get our genetic distance between F and OD. Okay, and that is, what was it? So we go to our single crossover 2, and that total is 25. Okay, cool. And that happens to be 2.5%. But like I said, it's directly proportional to your centimorgans, so, or your distance between the genes. So now we have our distance, 2.5 centimorgans between F and OD. Cool, so now we can actually construct our genetic map. So our gene order is G, F, OD. And I know for a fact that there's 11.7 centimorgans between G and F, so I just write it down. And then I know that there's 2.5 centimorgans between F and OD. So those are your distances. That's how you write out a gene map. That's how you do your calculations, only when you're dealing with single crossovers and no double crossovers. My next video is going to be on gene order with double crossovers. So that's going to be a little bit different, pretty much same concept. It's just a few new things that we'll do. So pretty much I showed you how to work out the gene order, and it's kind of like a process of elimination what I've done here. So when I was doing all that, I had to kind of check. I had to go back to my no crossovers, and can I get that progeny in my single crossover? Can I get that progeny in my single crossover 2? You know, so that's how you do it. Just remember it's a process of elimination, kind of like Cluedo. That's how I kind of think about it. But yeah, that's how you do gene order and genetic distances. And if there are any questions, let me know in the group or the comments. Cool, thanks guys.